Hello, good afternoon, everybody. Thank you, Annika, for the introduction. So I'm Niklas Tysklind, and I work in the mixed research unit of the ecology of the forest of French Guiana, which is in northern South America. And it's an overseas department of France, so I'm lucky enough to work in the European Amazon rainforest. As we all know, genetic diversity is essential for the potential of adaptation of all species to an ever-changing environment. And this genetic variation can be reshuffled by many different ecoevolutionary processes. However, the only way of getting truly original genetic variation is through mutation. In animals, early distinction between the soma, here in blue, and the reproductive germline, here in red, protect the descendants from somatic mutations, here represented by a yellow star. In plants, things are a little bit different, or not so clear. Somatic mutations occur in meristems along the branches, and they may accumulate to create slight variations in the genome of a plant. And these have been proposed to hypothetically be able to contribute to plant herbivory defense and plant immunity. However, it must be noted and stressed that these variations can also come from variations in gene expression, epigenetics, or viruses. So overall, mutations can increase, hypothetically, the individual fitness, but also they increase the intra-individual genetic load. If, like in animals, segregation between the germline and the soma occurs very early on, then the descendants will be protected from somatic mutations, like in Arabidopsis thaliana. If, on the contrary, reproductive tissues are made from the somatic tissues, then the mutations will be incorporated into the descendants, like in oaks. And this could have important effects, because some deleterious alleles could be incorporated into the descendants, having evolutionary consequences. So therefore, the timing of the segregation of the germline is of critical importance, because it will dictate the rate and spectrum of the heritable mutations, and then have profound ecoevolutionary consequences of trees. So, tropical trees are exposed to much higher UV rates than the temperate counterparts, so they could potentially have a higher rate of mutation. They are very, very large trees, so they have many cellular divisions, so again, they could have higher mutation rates than temperate species. So, one potential question is that temperate trees could have a potential for much higher mutational loads than temperate species. With this in mind, we set up for the tree mutation project, where we had a few hypotheses. So we assumed that somatic mutations follow the tree architecture, that increased exposure to UV light increases somatic mutations, that new mutations are rare in tissues, and that the trees accumulate very few somatic mutations. And finally, that somatic mutations are heritable to the descendants. So, to test this, we chose to work on two different tropical species, Dicorinia guianensis and Sextonia rubra, which are two of the most commonly exploited species in French Guiana. And our selection was based on the fact that they produce very large trees, that they have an architecture that allows us to test what we wanted to test, that they have an ecological and economic importance for French Guiana, and that we could predict flowering and obtain fruits, that they have small genomes. So for this, we tested 15 candidate species and estimated the genome sizes. And then we selected two individuals, which are called Angela and Sixto, for the rest of the presentation. So these were very large emergent trees. Angela, for example, was 52 meters tall, so very big trees, hard to climb. And first of all, we did 3D modeling or light transmittance with LiDAR. And this way we could estimate the light transmittance, the annual light transmittance throughout the tree canopy, and especially at the sampling points. We also validated this light influence by measuring with an Acupar device on spot. Then we characterized the whole tree architecture by measuring all the lengths of the branches and the diameters all across the tree, of all branches that were over 10 centimeters in diameter. to estimate the distances between, the physical distances between our sampling points. We then sampled the cambium at the base of the tree in 3 different directions to estimate the closest genotype to the embryo and identify mutations that are present from the base of the tree. We then selected pairs of branches which had branches of high exposure to light and low exposure to light, 4 in Sixto and 5 in Angela, where we sampled 3 leaves, which are biological replicates, so that we could distinguish sequencing errors from mutations. So our sampling design was these pairs of branches where what we were trying to do was to compare the effect of UV radiation depending on the branch lengths, comparing branches that were highly exposed versus lowly exposed. And we also sampled fruits to estimate the heritability of mutation and also try to assess the onset of the germline segregation. So first of all, we produced 2 very high-quality new genomes using high-fidelity reads and optical maps plus transcriptomes. We identified all of the transposable elements, and also, thanks to the transcriptomes, we identified genes. We had over 99% of the Busco genes of the green plant genes. And we had a very high coverage of 1,500x or 720x for Sextonia. We then we did sample resequencing of 33 samples in Angela and 27 in Sixto. Please note the high coverage that we used, which was higher than the average used in plants. And we then set off to detect mutations. So first of all, we masked all heterozygous sites based on the genome of heterozygous sites that were inherited from the parents. We then used the trunk mutations we used genomes from the trunk to identify mutations that were already present at the base of the tree, which we also masked. We then used leaf mutations, where we accepted only leaf mutations that were found in at least 2 different leaves. And finally, we resequenced these mutations in seeds. In terms of how we went for detecting mutations, so most plant studies have used SNP collars. And the problem with using SNP collars for identifying mutations is that they use 2 different tissues. They compare one with each other. And they have a tendency to overestimate, identify mutations when there isn't. So here, the problem with the low sequencing depth in one of the samples means that the program has identified mutation when there isn't. It's actually in heterozygote sites. And then it also has the opposite problem, where they fail to detect mutations when there is low sequencing depth in one of the samples. This is particularly important when the allele frequency is very low. So here with this diagrams here, I'm representing the frequency of the mutation within a tissue. So to correct that, there's the possibility of using cancer collars that compare both samples together and actually are better at correcting these 2 types of errors. So we did a benchmark analysis where we compared quite a few different allele collars, and we did a lot of simulations. And basically, we compared the recall capacity and the precision of detecting mutations. And we found that Strelka2, which is a cancer mutation collar, was the best collar, which we used to detect mutations. So we found over 15,000 mutations in Angela and over 3,000 mutations in Sixto, which is far higher than the average that normally was reported in previous studies. However, we also used the same analysis on previously published data from Oaks and Beach, and we found actually that the data was pretty similar. So we found a lot more mutations than previously published in Oak and Beach. It is interesting to note that if we consider only the allelic so mutations that are a higher allelic fraction, then we find a lot more mutations in the temperate species than in our 2 tropical species. However, when we consider all of the mutations in especially the lower allelic fraction ones, we find a lot more mutations in Angela and Sixto than in the temperate species, indicating that low-frequency mutations account for the vast majority of mutations within individual. So how are these mutations distributed? So we mapped the potential origin of all of these mutations based on which 2 pairs of leaves shared the same mutation. And we found that most mutations are originated at the base of the tree. I mean, we must think that these trunks are over 30 meters tall before the first branching event. So most mutations were based here. And as we approached the tips, we found fewer and fewer mutations that were shared among the 3 leaves. We then compared the branching topology with the distribution of mutations. So here in these graphs, we have the actual physical measurements of the tree and the branching length. And when we compared this to the phylogenies, we found absolutely no correlation. So how did we explain this? I mean, mutations seem to only partially be following the tree architecture. And this is probably explained by a mix of biological and technical limitations. So obviously the mutations, they may happen at one point in the stem, but they do not overtake the whole tissue. So obviously then by the mosaicism of the tissue, then the mutations might actually not distribute equally everywhere. And also the problems of low sequencing depth. In terms of what happened in terms of comparing the light exposure branches with the shadow branches, we found absolutely no significant difference between the number of mutations found in light exposed and shadow exposed, even when we started looking at specific mutation types. So here in this graph, we have the number of mutations on the y-axis and the light and shade branches. So we found no significant differences between no effect of sunlight on the number of mutations. However, it must be noted that because we had so few branch-specific mutations, then we cannot rule out an earlier effect of sunlight in the number of mutations in the tree. Then we looked into the distribution of the frequency of mutations, depending on whether the mutations were synonymous and non-synonymous, and whether they were in introns or intergenic. And we found a very significant effect of where non-synonymous mutations in coding genes were significantly rarer than other classes in Angela, but funnily enough, not in Sixto. This could be explained by the lower sequencing depth of Sixto, but also because we could not make a transcriptome for Sixto because of technical problems. So our gene model may not be as correct as in Angela. In terms of the transmission to the next generation, we tested 160 mutations. So in these graphs, we have the number of mutations on the y-axis and the allelic fraction on the x-axis. And in yellow, we have the whole the whole diversity of mutations. And in gray, we have the tested mutations. And what we found was that we found 6 mutations were transmitted to the descendants in Angela and 4 mutations in Sixto. But funnily enough, these mutations were not on the high allelic fraction, so they were not the most frequent mutations, but they were all very low allelic fraction mutations. So overall, mutations were transmitted to the seed, so we could expect to have a late germination, germline segregation. There was no high allelic fraction mutations transmitted. All transmitted mutations were at low or very low allelic fraction, and therefore lower frequency mutations play a significant role in the creation and maintenance of the standing genetic variation. So to conclude, mutation rates and mutation spectra are not demonstrably affected by differences in UV exposure. Mutation phylogenies deviate from the branching topology of the tree. Low frequency mutations are transmitted to the next generation, and low frequency mutations account for the vast majority of inter-individual variation. So, altogether, our results suggest far more complex links between plant growth, aging, UV exposure, and mutation rates that commonly thought. And more specifically, the transmissibility of low-frequency mutations, which are the most abundant, indicate a non-negligible role of low-frequency mutations as heritable mutations fueling plant evolution. This work is obviously the work of a lot of people, but I would like to highlight especially Sylvain Schmidt, which was our postdoc during the tree mutation project, and my co-PIs, Miriam Hoyerts and Thibault Leroy, with whom have been the core team of this tree mutation project. And of course, I would like to thank our funders, so the Centre d'études de la biodiversité amazonienne, and all of our different partners of this project. Thank you very much.
Thank you very much for a very interesting talk. We're a tiny bit over, but I think we've got time for a couple of questions, if anybody's got anything. I actually was wondering, you sampled the leaves, and I wondered how long they typically stay on the tree, and how do you know how long they've been on by the time you sample them? Because presumably that will have an impact in terms of how long they've had to accumulate mutations.
Well, because we only sample mutations that were present in at least 2 leaves. There were not mutations that were generated on the leaf. How long the leaf stays on the tree, I'm afraid I don't know the answer. But I mean, the mutations were obviously previous to the formation of the leaves.
Has anybody else got a question? Eva, yeah?
Yeah, I'll ask my question only if you promise to speak into the microphone.
Oh, sorry.
What kind of mutations are those who are transmitted to the next generation? The few that are transmitted to the descendants, are they synonymous, non-synonymous?
I don't know. Sorry.
Huh.
I didn't look into that.
How did you not look into that?
Sylvain, sorry.
Now, the reason why I was thinking about this is that we often tend to underestimate the fact that, for example, pollen is an organism of its own. So it has got gene expression. So whatever mutation you get in the pollen might affect pollen function. So maybe you could tell the climbers to do something more, even funnier. So go and collect the pollen in the flowers and see how it grows, and then sequence them and see whether they have the mutations.
Yeah, we did think about going and sampling the flowers. I mean, we can still go and do it for Angela and look into the mutations, but I mean, it was also a limitation of funding in terms of how many samples we could sequence. So we decided to go for the fruits instead of the pollen. But, and also there's the question of how to separate the different pollen grains and extracting the DNA from each and every one of the grains.
Yeah, another question?
Yes, thank you. Thank you, very interesting. So I was really surprised by the fact that the sun-exposed leaves were not having more mutations. Does it actually mean that they are somehow tumor resistant, so cancer resistant?
Well, what we found with our data was that we couldn't find a significant effect. I mean, it might be also a question of power in terms of the sequencing depth. I mean, we went quite far, but perhaps not far enough. Perhaps if we sampled more. And also, there's the question of, obviously, we were comparing branches that now are, we found them to be highly exposed or lowly exposed to light. But, and we tried to get them as close as possible so that we could get comparable rates. But yeah, maybe they do have mechanisms to protect themselves from mutations. And you know, our sample size is very small in terms of the comparison between tropical and temperate species. But you know, we did find fewer mutations in the 2 tropical species than the temperate species. So it might be that tropical species have better ways of coping with mutation. But obviously that's something that we would have to look into it further.
Okay, and I think we've got the last question from Santi. I'll give you.
Yeah, just curious if you had looked to where these mutations are in the genome, if they are randomly distributed or they are clustered in particular regions. So.
Yes, we did look at the distribution, and it's actually on the graphs, but I did not went into it. But no, the mutations are are randomly distributed for Sextonia, but not for Dicorinia. I can show you the graphs later, but I mean it's on the graphs. So the mutations are actually quite skewed compared to the transposable elements.
Thank you.
Thank you very much for your very interesting talk.